Okay, so the scientific news of this month. So from the US, there is some bad news coming. So Trump is threatening to take away the tax-exempt status of Harvard University. So the assault on US universities is continuing. The number of measles cases is also increasing in the US. Yeah, and then conferences. Let's go to aging, which is why we are here. So here in this study, they looked at animals with 107 mammalian species, to be correct, that had like 100-fold difference in longevity between these species. And what they did for all these species is they were looking at the genetic sequences for proteins, and they were aligning them and seeing where the differences were. And what they found is that in in long-lived species, some places where the DNA, the protein, I mean, is acetylated, you can actually have an amino acid like glutamine or arginine that actually mimics as if there is an acetyl group there, but there isn't one. So it's like an amino acid that acts as if it was a lysine that was acetylated. Some of these substitutions happen in both directions. So there where long-lived animals have it's a little bit difficult to explain. So in short-lived animals, there is a lysine there which can be acetylated, and then you have an acetylated protein which, for example, activates the protein, and then the same protein in the long-lived animal has, for example, an arginine there, which acts as if it's a lysine that is acetylated. And so in that case, because it's an amino acid change, it's always in the on state. I hope that explains it. And so that's what they discovered. And then they validated this for 2 proteins, I think it was, where they actually made these amino acid changes, and they showed that indeed it worked as in the longer-lived animals. Here they looked in humans at variations in the IGF-1 gene. So you know from my studies that if you modify IGF-1 signaling, you can extend lifespan. But until this point in time, they had never discovered any mutations in human populations in the IGF-1 gene. But here in as a Ashkenazi Jews, they actually found 2 variants in this gene. And both of these variants, the first one actually prevents or lowers the interaction between IGF-1 and its receptor, and the other one lowers IGF-1 levels. So both reduce IGF-1 signaling in different ways, but both reduce it, and both are found in these long-lived people and not in shorter-lived people. So this is a paper from Uri Allon. He's a very well-known systems biologist, but in recent years, he has been shifting his focus on aging. And so he's looking at interventions and the effect of interventions on the survival curve, and questioning whether they extend the sick span or the health span of an animal. And so that's what this paper does. And then we come to this paper from David Gems. And so he looked only in C. elegans here, and he used 3 different interventions. Temperature, he used an antibiotic, and he used multiple DAF2 mutants that are all long-lived. And he did all combinations of those. And then he looked at what the effect was on the survival curves. And he also looked at what the effect was on health span, which he defined as movement. And he comes to a bit of a contrary conclusion than anyone else, where he actually concludes that this alpha parameter, so the what we normally call the aging independent mortality part of the Gompertz curve, is actually aging related. Though you have to take into account that this aging related is based on the health span, sick span thing, and not And then the aging dependent parameter is not, according to him. So this is a bit contradictory to other researchers. And so in this paper, they looked at telomerase gene therapy, and they looked again at the Gompertz curves, and they basically found that probably a small number of processes were actually explaining aging. They say that mathematical modeling says that it's 3 to 5 processes with substantial crosstalk between them would explain the aging process. And if you could reduce the aging rate using the Swedish survival subdata, you could have a medium human lifespan of 85 from 85 to over 100 years. I assume that's with the based on this telomerase treatment, I assume, but for details, I will have to read the full paper here. It's not detailed. Here, it shows that people who have in breast cancer tissues, there is actually an accelerated aging in the methylation clock. But what is a bit weird is that actually in the same people, in cervical samples, which were non-carcinogenic, they actually had a decrease in epigenetic age. But what the relevance of that is unclear. That's technical, so I'm going to skip that. Yeah, here they look at changes in the T cell population with age and how there are specific changes in the amount of clonal expansion of cytotoxic T cells. And here they transplanted old and young hematopoietic stem cells into new mice. And what they found is that both the old and the young, I mean, hematopoietic stem cells were equally able to reconstitute the bone marrow of mice, and that it basically was not whether the cells are young or old that were transplanted, but whether the host was young or old. That actually explains the functionality of the hematopoietic stem cells. Here they actually took human stem cells, so pluripotent stem cells, and they modified them in such a way that they will no longer be recognized by the immune system, and so will not be rejected by the immune system, with the hope of developing a cell therapy that you literally could give to anyone. So it won't matter if you give it to me or you give it to you, Didier. You know that, of course, due to when you transplant cells or same as with organs, you need to normally match it to the recipient. But here they try to make it so that you don't need to do that matching, that the immune system will not recognize these cells. And indeed, when they transplanted these cells into mice with a normal immune system, the mice did not recognize these cells and did not reject them. And they showed that these cells, which they did in animals with Parkinson's disease, could actually reverse the motor problems, so the movement problems in the Parkinson mice. And these cells also had the suicide gene built into them, which is like a safety switch, of course. If anything goes wrong, you can kill off these cells. And when you killed off the cells, then of course the effect on Parkinson's disease again was diminished. So it showed that these cells were the ones that were responsible for the beneficial effect on Parkinson's. So this is kind of very promising, I think, development in cell therapy. Here they developed antibodies that block tau from being taken up by cells. So when a cell in the brain of a person with Alzheimer's that has tau proteins in it, when that cell dies, the tau proteins come free, and then those tau proteins can actually be taken up by other neurons, and then that is the way that this tau pathology can spread from neuron to neuron. And here you can use antibodies to block the uptake of tau, and thereby preventing the spread of tau from between neurons. Here they show that in people with Parkinson's disease, there is an accumulation of iron, but that was actually already known. So this is more confirmation of what is already known. Here they show that urolithin treatment. So if you recall, urolithin is this compound that is famous from pomegranates, that this actually improves alpha-synuclein aggregation in a mice model of Parkinson's disease. Here they developed these chimeric antigen receptor CAR T cells, which are targeted against amyloid beta, and they give these to mice, and they show that this then can remove amyloid beta in the brain. So rather than using antibodies, which is what is currently in humans already in the clinic, where you give antibodies and then antibodies bind to amyloid beta, and then phagocytic cells in the brain can take up the amyloid beta and destroy it. Here you actually administer cells into the brain that are clearing out the amyloid beta directly. And here they show that in older women, but not in younger women, and old is over 70 years of age, that hormone treatment, so for menopausal hormone treatment, actually accelerated the accumulation of tau, but that didn't happen in women under 70. So they say that probably the hormonal treatments for people over 70, for women over 70, might have to be revisited, the treatment guidelines. And so here they show that senescence of endothelial cells, so those are the cells that line the blood vessels, is an important contributor to the problems that we have in the vasculature in the brain. And so if you remove these senescent cells, you can improve the brain or the blood vessels in the brain, their function. So here they look in, it's only 5 people, older people, who were given dasatinib and quercetin. So these were people who had early Alzheimer's disease. And so they take some blood from these people, and they show that there is a downregulation of inflammatory genes that can be observed. But not, I mean, this is the only result of this paper. There is obviously you're looking at only 5 people. They cannot do much more than that, but you can see there is a decrease in inflammatory genes. And here, interesting, because Didier just mentioned at the beginning of the meeting of these extracellular derived vesicles. And here is a paper where they show that these vesicles actually have microRNAs in them that actually are xenomorphic. So they suppress the SASP in cells. And so you can actually take these. So what they did actually is they made artificial extracellular vesicles, just liposomes, and they included in those 4 different microRNAs, and they could use those to actually suppress the SASP.
So that means that, if I may, that means that you're actually reversing senescence rather than just killing the cell or something, correct?
Well, reversing senescence
anyway.
does not necessarily mean that the cells will start dividing, for example, again, but you are changing the expression, the secretion of these harmful inflammatory molecules that, and matrix remodeling factors, the whole SASP factors that's being changed. Okay. So yeah, here they use apigenin, which is a natural molecule that is also known to be a senolytic, and this improved the health in mice, and it also reduced cancer cell malignancy. Sorry. Here they used senolytic treatment, again, dasatinib and quercetin, the famous combination, to improve age-related hearing loss in mice. Here they show that this enzyme alpha enolase is important in senescent cells. Here you have the OS01 peptide in a topical formulation. and they show that when this was given to 60 female volunteers between 60 and 90 years of age for 12 weeks, there was an improvement. So here, for example, 70% of patients who were treated had noticed an improvement in general skin appearance versus only 42% in control. So that's just what the people themselves observe. But then they also show that systemic levels of pro-inflammatory cytokines were decreased in the people who were treated with that peptide and not in the control group. And they also demonstrated that the glycan age was also lower in the people who were treated with that peptide. Here, when you have these planarians and you cut a piece of them, they can regenerate the piece again. And what is interesting is that the piece that is regenerated, that piece itself is not just young, but actually the rest of the body, the old body, regenerates and becomes younger again as well, according to the aging measures that I used here. So they used multiple molecular signatures of aging, but you will have to read the paper to find the exact, I haven't read the paper, the exact signatures that they are talking about. Yeah, here they show that SIRT5 is important for the skeletal muscle function in aging. And if you have a deficiency in SIRT5, then you actually have an accelerated aging of the skeletal muscle. Here they show that inhibition of ferroptosis. So ferroptosis is an iron-dependent cell death program. And if you inhibit that, and there are multiple compounds such as ferrostatin 1 here, you actually improve lifespan and health span. So for example, here, when you treated C. elegans with FER1, so ferrostatin 1, you saw an increase in lifespan, and also an enhancement of motor function, tissue integrity, and a decrease in a decrease in cognitive decline, both in prematurely and naturally aged mice.
Does that, may I ask a question?
Yes, certainly.
Now, that ferroptosis, that includes the release of carbon monoxide. Is that correct?
I don't know that.
Okay, I think it does. That's, you know, any time that like those heme type molecules break down, they release a bunch of carbon monoxide. I wonder if it's related to the results. Go ahead, sorry.
Yeah, interesting question, but I don't know that. So, of course, heme proteins are known to bind CO very strongly. I mean, that's why you get serious problems if you inhale CO, because it blocks all the hemoglobin. But yeah, I don't know exactly what will happen during ferroptosis, if there is any change in CO secretion. So here, this is a paper in yeast, so it's a little bit very far from. But what they did is they actually inserted a protein in the cell membrane of yeast that actually allowed ATP from the external environment to be taken up by the yeast cells. And so that allowed them to actually just add some ATP into the medium in which the yeast cells were growing. And then by doing that, you increase intracellular levels of ATP. And when you did that, they showed that the lifespan of the yeast cells was increased. So here, this is a trial in humans with rapamycin, the PEARL trial, 48 weeks. It was double-blind, randomized, placebo-controlled, where people received 5 milligrams of or 10 milligrams of rapamycin. Now you have to be careful because they had some problems with the compounding of the rapamycin. So the blood levels achieved with that were much lower. So it was like, I don't know, what was it? Even the 10 milligram, I think, was only as if you were taking 3 milligrams or so of regular rapamycin. So, and the 5 milligram was even lower. So these people basically had very low level of rapamycin, even compared to what people normally take themselves who experiment on themselves for longevity. They typically take something like 6 or 7 milligrams of active real rapamycin rather than this lower efficiency version. So very, very low dose, but there were some positive effects. Well, very important to say is that there were no negative effects, so they didn't observe any side effects. They didn't find changes related to adiposity, visceral adiposity was not changed. The blood biomarkers were all remaining normal, so there was no problems there. There was a decrease in self-reported pain, which is good, and so in women who were taking 10 milligrams of rapamycin, it wasn't improved in men, and self-reported emotional well-being was increased, and general health was also increased in those who took 5 milligram. That's surprising. Emotional, why rapamycin will do that? That was something I will not expect, but that's what the data say.
And rapamycin is used primarily to suppress immune response. Is that correct?
Yeah, I mean, if you take it chronically, you will decrease your immune function. But for example, like I take rapamycin myself once a week, and I can say that when you look at my blood profile, you look at the number of immune cells, you look at the antibody levels, they are all remained unchanged. Because
how much do you take once a week?
6 milligrams once a week.
Thank you.
So the rest of the week basically allows my immune system to become normal again, But I mean, it's known to be xenomorphic as well. So you can lower SASP. So in old people, you will expect a decrease in inflammatory signaling when you take rapamycin, for example. Here, they show that rapamycin actually is beneficial against something called medial arterial calcification. So that's something that happens and that impairs the blood flow into the lower extremities in your legs, and that causes problems. And so rapamycin here in a mice model was beneficial against that. And here in mice, they were looking with rapamycin. This is only an abstract, so for a scientific conference. So when they gave rapamycin to mice, they showed that in male mice, right? Yes, in male mice, you had an improvement in the calcification of the joints, but there was no difference in females. But females anyway have lower problems with calcification than males. So the fact that it doesn't work in females is not as big of a problem. It works actually in those who have the biggest problem. And so that suggests that rapamycin could also be an interesting treatment for osteoarthritis.
Preventive or curative? Preventive, right?
Well, preventive mainly, yes. And so we already had a mice model, or a mitochondrial DNA mutator mice model before. So it's a mice model in which the polymerase gamma, which is the polymerase that copies the mitochondrial DNA, it has a defect in proofreading capability. So these mice accumulate lots of mutated versions of the mitochondrial DNA, and they have accelerated aging. But the problem is that until now, this happened in all the cells of the body, and it happened from the birth constantly. And now what they have designed is a mice model where it can be tissue specific and also time specific. So you can let these mice age naturally for a while and then induce the mitochondrial defect at an old age, or you can cause the mitochondrial problem only in selective tissues. So this will help into better understanding the role of mitochondrial mutations in aging. This is an interesting paper where they designed an AI way of identifying new geroprotective compounds, but more specifically compounds that work on more than one target at the same time. And so they actually looked at 5 different receptors, and they tried to find compounds that will bind to, I think it was at least three of these 5 receptors. And then the compounds that were found in that way were then tested in C. elegans. And over 70% of these selected compounds ended up extending lifespan, which is a very high success rate here. And furthermore, these compounds were in the top 5% of all geroprotectors in the drug age database in terms of the effect on lifespan. So the percentage increase, they had very high percentage increases.
I have a curiosity. How much was achieved?
I would have to look again, but I think it was something at least above 50% up to 70 or whatever percent, I think.
Oh, interesting.
Lifespan extension.
Interesting. Thanks.
Yeah, well, let's go here. Yeah, this is a bit of a warning sign. So they did prolonged fasting, but on the other hand, people who do it for longevity never do it this long because they did it for actually 10 days on average, which is quite an extreme amount of fasting to be doing. And when you do it for that long, then they observed actually that there was an increase in systemic inflammation. So yeah, maybe we shouldn't be fasting that long. But what they also observed is that there was a significant reduction in circulating amyloid beta proteins, AB40 and 42. So this actually suggests that it may be beneficial against Alzheimer's disease. But yeah, maybe you could reduce it to be a bit less extreme. This interesting paper links wealth and mortality in United States and Europe. As expected, the wealthier you are, the longer you live, both in the United States and in Europe. But the gap in the US is bigger than in Europe. And in fact, one of the most extreme things here is that the survival in the wealthiest US quartile was similar to that of the poorest quartile in Northern and Western Europe. So that's quite extreme here. So yeah. Here they show that if you replace butter by plant-based oils, you have a decrease in mortality rates. So you can look it here up 15%. So the highest butter intake was associated with a 15% higher risk of total mortality compared to the lowest intake. And the highest intake of total plant-based oils compared to the lowest was associated with a 16% lower total mortality. It was statistically significant for canola, soybean, and olive oil. And every 10 gram per day increase in plant-based oil intake was associated with an 11% lower risk of cancer mortality and a 6% lower risk of cardiovascular mortality. Whereas a higher intake of butter was associated with a higher cancer mortality, and substituting 10 grams a day of total butter with the equivalent amount of a total plant-based oil was associated with an estimated 17% reduction in total mortality, 17% reduction in cancer mortality. So yeah.
Doesn't it matter how much oil? I mean, if I'm drinking a quart of olive oil a day, you really think that's going to help me?
Sorry to stop, but it is the 5th time that you are asking questions now, and also one person has to leave pretty early today, so I propose to stop with the questions.
I'm almost done with the scientific news. At everything, there is an upper limit, Walter. So drinking a half a liter or a liter of olive oil a day is probably not the best idea. And I think you will get some diarrhea from that as well. Here, actually, outdoor heat in the United States was, so if you have higher outdoor heat, so just heat waves, was associated with accelerated epigenetic aging. So I'm accelerating a little bit. Here they show that particular matter in China, a higher level of that was associated with a decrease in cognitive function. So basically air pollution here. That's a paper by Aubrey de Grey on the RATE database, but there is not much to say, it's just a database. And here they show that photoaging clock that they developed and it predicts all-cause mortality among occupations. We skip C. elegans. Reviews. This is a nice paper you might want to take a look at. And I think that's it. Those are all just reviews. So, okay.